Hi, okay, so today we're going to start talking about reproduction and we're going to have two parts. So this is going to be development and male reproductive, and then we're going to, the next section will be female reproduction. So I still have the abbreviation references and the endocrine glands, all of those for reference. And so today, so I circled these here. Today we're going to be talking about um, some new hormones, and they're the hormones that are released from the hypothalamus and the pituitary. So it's going to be that HP axis, the hypothalamic pituitary axis. So the in this case, it's a gonadotropin releasing hormone will cause the release of two other pituitary hormones, which are follicle stimulating hormone, that's what FSH is, and luteinizing hormone, that's what LH is. So follicle stimulating hormone, the name of that comes from in the ovaries, there are developing follicles that will then become the oocyte which is the I guess that's what the sperm joins, and that's what makes the baby, is that egg. Okay, and so that's the follicle when it starts out, and so FSH in a female will stimulate that follicle to start, for the menstruation cycle and ovulation. Luteinizing hormone has to do with also something in the ovary. It's called the corpus luteum, which is what develops after ovulation. So, and it's corpus luteum just means a yellow body, and that's because that's what it looks like. So these names, follicle stimulating hormone, luteinizing hormone, they make sense, and they're named after the female, the processes that take place in females in the ovaries. So some of the other hormones that we're going to talk about: the testes produce testosterone, the ovaries produce the estrogens and estradiol and progesterone. Okay, so these two things, and we're going to get into that and how those change and matter. And then estradiol, progesterone, also formed from that corpus luteum that we talked about. And then the placenta also produces some of these: human chorionic gonadotropin, and then placental hormones. Okay, which we'll cover from in the female productive system. But today, now we're going to just talk about these: the GnRH, FSH, LH, and the testosterone. Okay, so how do we define someone's sex? When we say someone is a certain sex, if they are male or they are female, it can get complicated. Usually, in most humans, then it's pretty straightforward. But there can be issues that come up that can kind of change things. So we have our genetic sex, and our genetic sex is either going to be XX, which is female, or XY, which is male. Okay, so we have 23 pairs of chromosomes. Or 23 pairs, yes. So we have a total of 46 chromosomes. We get 23 chromosomes from dad, and we get 23 chromosomes from mom. Okay, and so you can see these are the pairs of chromosomes here. So 1 through 22 here, and then, oh gosh, this is so tiny, but you can see that this one right here, that's the X and the Y. You can see that the X is very large and the Y is a much smaller chromosome. Otherwise, the other 22 autosomes, the other 22 chromosomes that are not sex related, they're not the X and the Y, those are pretty much identical. They just have a different gene sequence, which will make proteins slightly differently, because that's all the chromosomes do, is they just code for proteins. But the X and the Y can have a big difference, and that's what we're going to talk about. Okay, so yeah, 22 are pretty much the same, these autosomes, and then you have one pair of the sex chromosomes, and that's what will cause your genetic sex to be either male or female. Okay, so normally that will translate to the next one, which is gonadal sex, which is the presence of male or female gonads. So if a male will produce testes and a female will produce ovaries. Okay, and so those are the gonads, the testes or the ovaries. And then there's phenotypic sex. Phenotypic sex is defined by physical characteristics of the internal genital tract, so internal genital tract, and then external genitalia. Okay, so what this kind of all implies is that, you know, you could have a genetic sex XY, but if something happens, you may not produce testes or ovaries properly, or it may not match. And then you could have testes and ovaries, but you may have different either internal or external genital organs or components. Okay? All right. So, genetic sex determination. So, gender is determined by genetic inheritance of 2 chromosomes known as the sex chromosomes. Okay, so we have a picture of this here. We can see the female XX, male XY, which means that when the female is going to produce an egg that's just going to have one. It's either going to have this X or this X, and it doesn't get really any other options. However, in a male, the male is going to donate either an X or a Y. So it is the male parent that determines the sex, the genetic sex of the offspring. Okay, if the male donates a Y, then it's going to be a male offspring. If the male parent donates an X, then it's going to be female, because the female will always donate an X because she's XX. All right, so X-linked genetic diseases and traits may be expressed in a dominant fashion in males because they only have one X chromosome that they got from their mother. So an example may be, let's say, usually it'll be like the X that's the carrier, and so let's say that there's color blindness, okay, in this family, and let's say that mom happens to donate an X for color blindness. Okay, so this is the X for color blindness, and so she just donates that one. She had a 50-50 chance. That's the one she donated. If it goes to a female, well, all that color blindness is that those little cones in your eyes that detect color, certain colors, wavelengths of color, maybe it just doesn't work right. Okay, so it's not going to detect, be able to distinguish red and green, for instance. There are a lot of different kinds of color blindness. But let's say it can't distinguish red and green. So in a female, if dad donates an X, well, then it's fine. She's not going to express that. She'll carry it, but she's got a healthy X here. That's fine. Okay, so even though she got this donated from mom, she's not going to be colorblind. The female offspring is not going to be colorblind because she has a healthy cone protein that she's making because dad donated his X and it was healthy. Okay, does that make sense? However, that same colorblind X donated to a male offspring, so here it is, okay, and dad donates a Y because it's male, so the male gets the XY, then that offspring, that boy, will be colorblind because it's got the colorblind gene, and there isn't an X to cover for it. There isn't an X to kind of make up for it. Okay, so that's kind of the advantage of having 2 chromosomes anyway, is that if one is defective, then it will the other one can cover for it, and so that's going to be like recessive genes. The other problem, though, that sometimes can happen is that one of the genes will make a product that is detrimental or causes disease, and that would be dominant, because if it's making something, then the other one can't stop it from making it. So I don't know if that makes sense, but anyway, that's kind of how genetic diseases work, and that's one of the features, I guess, of having a chromosome from mom and from dad. Okay. All right, so sex differentiation. So there is a chromosome, on the Y chromosome there is a gene. So a chromosome is just a gene. It's just part of the chromosome that expresses a certain protein. Okay. Okay, and one of these is called the sex determining region of Y. So if this is the Y chromosome, on that chromosome is a gene called the sex determining region of Y, and it produces a protein that then goes on to turn on a whole lot of other proteins, okay, on different chromosomes all throughout the genome, the body. Okay, So during the 7th week of fetal development, that sex-determining region of Y is expressed. It suddenly turns on and it starts producing this protein, and then this protein causes a lot of other stuff to happen in that fetus, that developing fetus. Okay, so before that, and so that's what I guess what it's turning on is it's turning on all of these proteins to start turning this child into a boy. Okay, so into a male. All right, so before that, the fetus is considered bipotential. It's neither male nor female, but when that SRY gene is expressed on the Y chromosome, which is where it's supposed to be, then it will start turning on all of the mechanics to make a male. Okay, so this SRY turns on the genes for male development, turns off the genes for female development. With no SRY expression, the fetus will then just go on to become a girl. So that doesn't mean that, you know, everybody starts out as a girl, which is a lot of times what people say. People start out as both or neither, really neither. And then when that SRY gene kicks in, it will turn it into a boy. If it doesn't kick in, then the fetus will go on to become a girl. All right. So internal sex differentiation. So what happens when that SRY is turned on? So development of the testes is stimulated. So that's the first step. Okay. So by the presence of that SRY. So the sex determining region of Y, the gene product goes on to turn on a whole bunch of other genes, and then those genes will start this process. So the first thing, if we're ordering them, is the development of testes. Now you can see up here in this picture, these are the early gonads right here, these blue blobs right there in this picture. Now those don't know whether they're going to be an ovary or a testis. Okay, so they could be either one. They could become ovaries, they could become testes. It is the presence of the SRY gene, after expression of that gene, that these will go on then to become testes. Okay. Now, so testes produce testosterone. So testosterone secreted from Leydig cells. So you're going to have to know these cells. Okay. Leydig cells produce testosterone in the developing testes, stimulates maturation of Wolffian ducts into the vas deferens. Okay. So the vas deferens is just something that carries sperm. We're going to look at it and what it does. It's shown in blue right here. Okay, so this is the vas deferens, and it kind of attaches to the testes, and then carries sperm to be, excreted through the urethra. So, the presence of testosterone will stimulate this thing called the Wolffian duct. See it up here? It's in blue. It will tell that duct to become, in this early developing bipotential fetus or embryo, to become to the vas deferens, but it's really all of these parts of the tubule. Okay, so yeah, it will cause that to differentiate. If this went on to become a female, that Wolffian duct would just wither away, which is what we see over here on the right. So on the left, we see it developing into the vas deferens, the epididymis. Okay, so it's developing, and then in the female, it just kind of is breaking apart and it goes away. All right. So for females, okay, so there's something over here. So if we stop looking at the Wolffian duct and we look down here at the Mullerian duct, then in females, the Mullerian duct will go on to form the fallopian tubes and the uterus. See this pink down here. Okay, so it'll go on to form that. Okay, and that's going to be part of the internal female sex differentiation. However, in males that are expressing the SRY, they will start producing something called anti-Mullerian hormone, or Mullerian inhibiting substance, MIS. Okay, so and if they do that, this is produced by Sertoli cells. Okay, and if males produce the anti-Mullerian hormone, then this Mullerian duct, okay, you can see it here in pink, will go on to, well, do nothing. It disintegrates. Okay, so you can see it kind of going away here, and then it's gone. All right. So that's the big difference. Those two things, the Wolffian duct, you'll have to remember that matures into the vas deferens. Okay, we'll say that. And then the Mullerian duct will go on to develop into the fallopian tubes and the uterus. Okay? So obviously the SRY wants to, that's going to cause that Mullerian duct to disintegrate, and then it will cause the Wolffian duct to turn into the vas deferens. All right. So, anti-Mullerian hormone secreted from the Sertoli cells. You also have to know the names of these cells. The Leydig cells are producing testosterone, and Sertoli cells are producing the anti-Mullerian hormone. And they do a lot more than that we're going to see later. All right, so growth and development of external male genitalia depend on the conversion of testosterone. I just have androgen here, but this androgen is testosterone. Testosterone, okay, to DHT, dihydrotestosterone. Okay. So in order to go from just being a gonadal male, okay, so you have the testes are there, the testes are making testosterone, you have to convert the testosterone into dihydrotestosterone, DHT, that happens in the testes. You have to convert that to the testosterone to DHT, so that will continue the formation of external genitalia. Without DHT or without the receptors for DHT, then the male genitalia won't develop. So that gives us our gonadal. So we have a gonadal male, but to become, to express that phenotype, to become the phenotypical male, then all of these things have to happen. Okay, so the testosterone to dihydrotestosterone to produce the genitalia. All right, so, and I have that note here that androgen receptors are required for that process, otherwise it won't happen. If there are no receptors for the DHT, even if you're making it, it's not going to do anything. Okay, so without SRY, gonadal females develop ovaries that produce estrogen, but because they don't secrete the anti-Mullerian hormone or the testosterone, then they will go on, as we see here, the Mullerian duct will go on to develop into the fallopian tubes, and that Wolffian duct won't, there's no testosterone to support its development, so it'll disintegrate. Okay, does that make sense? So the one or the other is going to survive. One is going to disintegrate, one is going to develop. It really depends on the presence of testosterone to develop the Wolffian duct and the presence of anti-Mullerian hormone to cause the Mullerian duct to disintegrate. Okay, so in males, so that's what we're talking about first. Descent of the testes sounds like a really bad book. So during the development, the testes form in the abdominal cavity, so that's where it starts on when it's a bipotential, I guess, phenotype. I don't know, but when it's neither male nor female, before development starts to take place, and then after development starts to take place. But anyway, it all starts up here in the abdomen. And then the testes move to the outside of the body because sperm production requires lower temperatures. So the testes will move out, and you can kind of see the testis here. And then it moves down, and it passes through something called the inguinal canal. Okay, so that's what you can see here on this picture, through the inguinal canal. And then outside, and it will land here in the scrotum. And then the inguinal canal, so it had to open up to allow the testes to move through. Testes, both of them go through, and then it has to close up again. Okay, so this inguinal canal. So although the path closes up, it creates there's a weak point in the lower part of the abdominal. So remember, this is of the abdomen. So remember, this is where your intestines are, and so. If you have a weak area there, what can then happen is if there's any extra pressure or something, then your guts, your intestines can kind of push through there and move into that area, and that's an inguinal hernia. So that's what's tested. You just kind of have to feel up in this area to see if you can, with any kind of pressure. This is like turn your head and cough. Any kind of pressure, if you can feel something trying to push through that, through that inguinal canal. Okay. All right, so summary of sex differentiation. So everything we just went through, a couple of pictures on that. So recall there are two major cell types in the testes that direct sex differentiation. So we have to know the cell types. The Leydig cells produce testosterone, and the Sertoli cells produce that anti-Müllerian hormone. Okay. All right. An LH, luteinizing hormone, stimulates Leydig cell activity, and FSH, follicle stimulating hormone, stimulates Sertoli cells. And that'll happen later on. All right, so, but we have to kind of keep all of those things straight. So Sertoli cells produce that anti-Müllerian hormone, or also called Müllerian inhibiting substance, and that's what it's called in this picture, so absence of MIS. Okay, so we can kind of go through that and kind of review all that. In the absence of these molecules, the fetus continues to produce a female. Okay, so everything we just went through, SRY gene, Sertoli cells make the anti-Müllerian hormone, Leydig cells make the testosterone. Testosterone causes conversion or the development of Wolffian ducts. Anti-Müllerian causes the Müllerian ducts to go away. Without that, in the female, the Müllerian ducts will develop into the fallopian tube's uterus and inner vagina, and the Wolffian ducts will go away. Okay. All right. So this is just another flowchart of the same thing. So disruptions in sex development. So there are often disruptions in the development process. Because remember, we have basically three different types of sex. We've got the genetic, gonadal, and then the phenotypic. And so if anything happens along the way, you may have one without the other. Okay, so there are a few examples here. There are others, obviously. So loss of function of Y. Okay, so if the Y does not function the way that it's supposed to, so the SRY gene is not expressed, then even though there is a Y gene present, it's not going to have, it's not going to produce the male. It's not going to do that, trick with the anti-Müllerian and the testosterone for the Wolffian duct. So it's a genetic male because it's XY, but it will not produce testes, so it's not even a gonadal male. It's just genetic. Okay, so there's also kind of along the same lines, but instead of being XY, XX, displacement of SRY to the X chromosome. So now we end up with, instead of the Y chromosome, there is no Y chromosome, but there's an X chromosome that probably a mistake or something that happened during development caused the SRY gene, remember the sex determining region of Y, to suddenly be on the X chromosome, which I guess technically would now make it a sex determining region of X. But either way, it's being produced, which means that genetic female X chromosome will go on to be a male. Okay, express as a male. Okay, so they'll have the gonads, and they'll have the and everything else that goes along with it. So sometimes there are mutations with partial function, so ambiguous genitalia, that there are other processes along the way, other gene expression proteins that are made that if they're not made properly or exposure to hormones can cause ambiguous genitalia. Okay. All right, so if a gonadal female is exposed to androgens in utero, remember it is the presence of testosterone that causes the Wolffian ducts to develop. Okay, so if a gonadal female is exposed to androgens, the Wolffian duct doesn't care, where it comes from. So when these external genitalia are differentiating, then a male phenotype results. So that could be before. If it's before the external genitalia are differentiating, then it's going to be male. That it's going to be a male phenotype. If it occurs later, so let's say that everything has already kind of developed or begun to develop, then a female phenotype is retained. So it may be, they may have, let's see, they may have more, I guess, masculine traits. Okay. Okay, so it may be seen with excessive androgen secretion from the adrenal gland or from the ovaries or from hormone medications. Okay. So this is something that's actually been studied quite a bit, and the timing of this is one of those things that's been studied, but a lot of sexual orientation, those kinds of things have, they're hypothesized that this may be something that may contribute to that, is just exposure to, it says from the adrenal gland. Remember, the adrenal gland makes androgens. The ovaries make testosterone. It has to make testosterone or other androgens to make estrogen. Hormonal medications could cause that, and like I said, if it happens before or while the external genitalia are differentiating, then this could be a phenotypic male. Or if it happens afterwards, then there are other things that could happen. All right, so the absence of androgen receptors in a gonadal male. So remember, we said that testosterone had to be converted into dihydrotestosterone, or DHT, for external genitalia development, okay, for the penis and scrotum. If there are no receptors, which is something that has been seen, okay, so this is something that happens. If there are no receptors for the DHT, then it's like the DHT was never made, which means that male external genitalia won't form, but female will. Okay, so this is going to be a phenotypical female. Okay, so even though it has gonads, gonadal male, phenotypic female. Okay. All right, so hormonal regulation of reproduction. So we mentioned early on that this is going to be part of the HP axis, the hypothalamic pituitary axis. So the hypothalamus is what produces the gonadotropic releasing hormone. Okay. So gonadotropin releasing hormone. And then the anterior, remember the posterior just makes vasopressin and oxytocin, so most of these are going to be anterior, and that's true for this too. So the anterior pituitary is going to produce the luteinizing hormone, the LH, and the follicle stimulating hormone. So remember, luteinizing hormone activates those Leydig cells, and the follicle stimulating hormone is going to stimulate these Sertoli cells. Okay. All right, so and that's made by the pituitary. The gonads, the testes, and ovaries produce steroid and peptide hormones. And so that's what's going to be stimulated are the testes and the ovaries. That's where the Leydig cells are. They're in the testes. And then we'll come back to this when we talk about the ovaries later. But also those. All right. Different cell types, though. Okay, so Leydig and Sertoli cells are specific to the testes. So the hormones that are produced by the gonads will feed back. So that's going to be negative feedback. To the pituitary and to the hypothalamus to inhibit release. Okay, so the feedback can be selective for specific functions, and what we're talking about here is that Sertoli cells can produce something called inhibin, whose sole purpose is to kind of shut down Sertoli cells. All right, so we're gonna talk about that in a minute. Now, here's a note here. Continuous release of gonadotropin releasing hormone will actually decrease release of the gonadotropins, LH and FSH. So what tends to happen is that it's kind of pulsatile. The release during the reproductive years after puberty, we're going to see that in a minute too, is pulsatile, and that's required for the release of these gonadotropins, the luteinizing hormone and the follicle stimulating hormone. So yeah, synthetic forms of GnRH are used as a treatment for androgen-sensitive prostate cancer, which is going to basically release or decrease LH and FSH release. Okay, so what that means is just what it says. If you give, if you provide GnRH, you're going to decrease the LH and FSH, and that stimulation can be increased or reduced. We'll also come back and talk about this when we talk about birth control, okay? Because that's kind of the pathway that's used for that too, but it's more of a feedback mechanism. All right, so puberty. So puberty, we'll go to puberty, and then we'll finish with the other process. So puberty is lots of things happen. So many reproductive and developmental processes. So I think most people, everybody in this class has been through puberty, so we kind of know what happens. So many reproductive and developmental processes, including puberty, are directed by the hypothalamic pituitary axis. So what physiologically happens is that the gonadotropin releasing hormone, the GnRH, will start to release this in a pulsatile way, which is going to cause luteinizing hormone and follicle stimulating to also release in a pulsatile way. So this is kind of what happens at puberty, is this is going to increase. So secretion of GnRH begins at gestational week 4. Okay, so if we look at back in development, that the secretion of GnRH begins at week 4, but the levels remain low. Okay, so we have this low level of GnRH. LH begins, let's see, gestational weeks 10 through 12, so still before birth is when that happens. Levels are also low. Okay, so LH is low, or GnRH is low, LH is low, and follicle stimulating is low. The release patterns change during puberty. So during puberty, they start to become pulsatile. Okay, and then they let's see, yeah, and throughout the productive years. So then, yeah, it's not until later in life. And I'm going to stop trying to draw that and just show you the picture that is nicer. Okay, so that pulsatile secretion, that's what starts at puberty. So the GnRH levels go up, and then you have the pulsatile release. So pulsatile release of GnRH begins at puberty, and consequently also the LH and the FSH begin, and that continues through the reproductive years. So upregulation, okay, in GnRH receptors. Okay, so remember, hypothalamus to the pituitary. Okay. That means in the pituitary there are receptors for the GnRH. So the GnRH is released. RH is released from the hypothalamus, and then it has to land on a receptor in the pituitary. So upregulation, so these receptors will increase, which will increase the release of LH and FSH. So upregulation of GnRH receptors in the pituitary increase the sensitivity. Okay, and so that's going to heighten the effect even more. So increased FSH and LH increase the production and release of testosterone. So testosterone is going to be from those Leydig cells, from the LH, and estradiol also from the is from, well, we'll talk about where that's formed. It's released from the follicle. Okay, so testosterone and estradiol are responsible for the appearance of the secondary sex characteristics associated with puberty. The onset of puberty has been shown to be genetically programmed, so that means if you're usually for menses, for menstruation, when that begins is usually very similar to the mother. If you were a late bloomer or an early bloomer, or if your parents were early bloomers or late bloomers, then that means their children will also, which I guess is you, will also either be a late or an early bloomer. So it's not obviously like anything that's genetic, it's not guaranteed, but it does show a genetic relationship. Okay, so age of onset is similar for parents and their children. The mechanisms that trigger the onset of this pulsatile GnRH secretion is unknown. We don't know what causes it. There are a couple of ideas here. Nutritional status may alter the process. So we say that because if a young person is under tremendous stress or caloric restriction, then there are delays in puberty, the onset of puberty that's seen. So caloric restriction delays in girls, that's been studied, and their development and their puberty will be later. Also interesting, melatonin. Okay, so there may be a relationship to melatonin, because melatonin is released from the pineal gland. So the pineal gland kind of sits up in your brain right above the optic chiasm. So it kind of sits up in your brain. Your eyes are up here. Okay. And it's pea-shaped, pea-sized, and it releases melatonin, and melatonin is involved in sleep. Okay, so increased melatonin will cause you to feel tired, but the thing is that melatonin declines through childhood and into adulthood. You tend to sleep less, and so the thinking is that, well, if it's decreasing, maybe it decreases to a certain point, and that's what triggers the onset of. Now, the mechanics are not known, the mechanism isn't known, but maybe that's what triggers this decrease in melatonin release over time. It triggers puberty. The other thing that makes studiers, researchers think that is that if you remove the pineal gland, it will precipitate early puberty. So if you remove the pineal gland in someone who is very young and hasn't gone through puberty yet, then that may trigger puberty. Okay, so it kind of is a decent indication that there's at least a link to the pineal gland and melatonin. All right, so characteristics of puberty. So these secondary sex characteristics are kind of located over here. So male, pubic hair, penile growth spurt. So that's going to be the same male and female. Pubic hair, that's also the same male and female. Spermarchy, which is males will start making sperm, and females will start ovulating. So menarche. Females also breast development, pubic hair, menarche, and everybody grows. All right, so what happens? Events are initiated by the pulsatile secretion of GnRH, FSH, and LH. Okay. So GnRH deficiency will delay puberty. So we know that that's a relationship. However, if you just have a GnRH deficiency and you say, okay, well, we're going to give you extra GnRH. Okay, so we're going to inject this. We're going to give you some long-acting GnRH and just give it to you every now and then and get your levels up. That's going to fix it, but it doesn't. So treatment with exogenous pulsatile GnRH will work. So if you just give GnRH to get the levels increased, that's a no-go. That's not going to initiate puberty. But if you give it in a pulsatile way, then that will initiate puberty. Okay. So treatment with exogenous pulsatile GnRH will initiate puberty, but the long-acting won't. Okay, so that pulse, even though it's not completely understood, obviously is very important. All right, so pubic and axillary hair is due to increased production of adrenal androgens. Okay, so andronarchy. So some people say that andronarchy, so that adrenal adrenarchy from the adrenal gland is part of, so that increases early or before puberty, could be an indication that puberty will be taking place, but that's going to trigger the pubic and axillary hair. Axillary is under the arms. Boys, Leydig cell proliferation, which is where testosterone is made, and this happens in the testes. Okay. The growth of the testes, so increased number of seminiferous vesicles. So we're going to look at those when we talk about sperm production, and that's where sperm are made. And growth of sex accessory organs, the prostate, and some of the other stuff that we're getting ready to look at. Girls' onset of puberty is earlier than boys. Okay. Budding of breasts occurs, followed approximately 2 years later by menarche, which is the onset of menstrual cycles and ovulation. And then estradiol secretion, which is the estrogen that's secreted during this time, is secreted from the ovaries, and that's increased, okay, for girls. Okay. All right, so let's go ahead and dig into the male reproductive physiology. So spermatogenesis, that is the purpose of the reproductive system in males, is to make sperm. In females is ovulation, the production of eggs and ovulation. So spermatogenesis is the production of sperm. So sperm production occurs in the testes, and then emission, and so it's made in the testes, but then it has to leave, emission and ejaculation through the urethra of the penis. And then a lot of other stuff. Hormone production takes place in the testes, which is associated with sperm development, but also some of the other characteristics that we've talked about. Okay, so now we're going to start talking about these cells and kind of trying to get our head around this. So I tried to put pictures in here that can kind of help, but this is a seminiferous tubule. Okay, so this is, the seminiferous tubule is a tube. Okay, so it's kind of, like this. If you're looking at it in a cross section, so this is looking at it like this is the lumen, this is the inside. So if this were here, it would be like this big, and the lumen would be kind of in this area. Okay, this is just kind of a chunk out of it. So if we look at these cells, so we have Leydig cells that are also called sometimes interstitial cells, and those are kind of out here. Okay, so we can see these, color a couple of them in. These are the Leydig cells. And those are producing testosterone. And you can kind of see they have access to the blood, so testosterone can move into the blood. But they also kind of diffuse into, well, they move through the Sertoli cells, actually. Let me see. The Sertoli cells are not really good in this picture, but these are the Sertoli cells, kind of in this area. Okay. Okay. These are spermatids. The blue ones, these are spermatids, spermocytes, primary spermocyte, spermatogonium. So this is kind of their development as they kind of move along, and they go through mitosis, meiosis, and then they become spermatozoa, or these sperm cells here. Okay, so it kind of moves from the basement membrane when in development, and then kind of moves down here, and then goes into the lumen of the seminiferous tubule, and that's where the sperm go. Okay. All right. So, Leydig cells, what do? Primary role, testosterone. Okay. Activated by LH, luteinizing hormone. Leydig cells produce testosterone. Sertoli cells, primarily activated by FSH. Those provide nutrients for these developing sperm. So we see all of these developing sperm here. And they don't have access to the blood. Okay, so there's something called the blood testes barrier, and so all of the nutrients that these developing sperm get have to come through the Sertoli cells. So the Sertoli cells pretty much are responsible for making the sperm. They provide the nutrients. They're also called nurse cells in some cases, or sustentacular. So they have to get the nutrients so the sperm can, and all of the factors that have to be present for these sperm to develop. Okay. All right, so that's the Sertoli cells, and that's sperm development. And then the developing sperm will move into the lumen of the seminiferous tubules, and that, as you can see here, is what makes the mass of the testes. So that's really what the testes are, or they're little carriers for these seminiferous tubules, that all the way through these things, sperm are being made all the time. Okay, all the time throughout the life of a man. Okay, typically, without any other problems. Okay, so what is spermatogenesis? We're gonna try to keep this as simple as possible, but it is it's the production of sperm, obviously. But there are three phases. There's a mitotic phase, which is where you just sort of form these germ cells that are going to become sperm. Okay. So this is just normal mitotic division. So these have 46 chromosomes, 23 pairs, just normal mitotic cell division. Well, then you have meiotic cell division, and this is where the chromosomes kind of separate, and some go into one cell and some go into the other. So now you have the situation where you have two different cells, but each have 23 chromosomes instead of 46. Okay, so remember there used to be two, there used to be a pair. Now there isn't a pair. So you're going to have one set. Crossing over has taken place, so that means they've shared a little bit of their genetic information with each other, and so now you have these two cells with 23 23 individual unique chromosomes. And from a male, it will either have an X or a Y. So it's going to have 1 through 22 for sure, and then it's either going to be an X or a Y. Okay. All right, so these are unique. They're what we call haploid, or N instead of 2N. They are 1N. And they can't really function as a regular cell. The only thing that they can do is look for an egg that also has 23 chromosomes and then fuse with that to make a new organism with 46. Okay. All right, so spermiogenesis, the last step. So that's so mitotic, just division to make a whole bunch of them to work with, and then meiotic. Okay, let's make our sperm with the 23, with the our half that we donate, and then spermiogenesis is that last step. The last step is the loss of the cytoplasm. So this you can see here. These actually still look like cells, but then you have a sperm. A sperm doesn't really look the same. Okay, so it's got a tail, it's got the head, and it loses that inside, the cytoplasm that's inside. So loss of that cytoplasm, formation of the flagella and the flagella tail, and that final maturation. So all of this stuff is taking about 65 to 75 days to make a sperm. Okay? But you're making 100 to 300 million each day are completed, and you saw all of those tubes that are in there of the seminiferous tubule. So it takes 65 to 75 days to make them, and then they start to move out of the seminiferous tubule. They move into the epididymis, okay, and then they just stay there for several months until they get their chance to be released. Okay. So normal sperm production requires a temperature 2 to 3 degrees Celsius below core body temperature. And so that's why the testes are kind of outside the body, to keep it below the body temperature. This is probably for related to infection and avoiding infection, kind of keeping the body temperature different. But there are muscles, cremaster and dartos muscle, that can contract and relax to pull the testes up if it's cold, or let them go down if it's hot, to kind of keep that temperature regulated. All right, sperm structure. I like how sperm structure names are made. So there's the head, which is up here, and that contains, so that's going to have the nucleus, and that's going to be carrying around the 23 unique chromosomes, and either an X or a Y, but not both. Okay. All right, so that's the head, and that carries the nucleus, and then there's something called the acrosome on the head, and that carries enzymes, okay, and other products to kind of fill with, to penetrate the oocyte, which is the egg. Okay, so that binds, and then these proteases, enzymes, are kind of used to kind of break down the egg. So then the chromosomes can go in. Okay, we'll talk about that again later. All right, so and then there's the middle piece. That's fun. There's the head, the middle piece, and then the principal piece, which is bigger, and then the end piece. Okay, so pretty straightforward. But the middle piece contains the mitochondria. And so the mitochondria does what mitochondria do, and they make ATP. Okay? And the ATP that's produced can then power the flagella. So this is the flagellum, and so it is going to, so remember, the flagella kind of moves around, and that's what propels the sperm forward. Okay. All right, so the tail, which is the flagella, the neck contains the centrioles forming the microtubules that comprise the remainder of the tail. The middle piece has the microtubules. The microtubules is what the tail is made of. Okay, and that's just a bunch of tubules that are able to kind of move. Okay, Okay, without getting into too much cell biology. All right, so yeah, the mitochondria has the ATP for energy, the principal piece, longest portion, end piece, terminal tapering portion of the tail, and then once ejaculated, sperm do not survive more than about 48 hours. This kind of varies, depends on which study you're looking at, but that's a kind of a ballpark. Survive more than 48 hours in the female reproductive tract. So around that. All right, so steroid hormones in the gonads. So before we move on, this is something that we kind of need to talk about. We're saying that we're talking about the sperm and spermatogenesis, and one of the things that we need for production of sperm from those Leydig cells, remember, is we need testosterone. Okay, so I wanted to talk about how these hormones are made and how they're related to each other, I think. So cholesterol, they're made from cholesterol. Remember, cholesterol is kind of that based on 4 rings, and it's a lipid. So these are all lipid soluble, so they can kind of move through membranes. They're not polar, they're non-polar. So it starts with cholesterol and then pregnenolone. Okay, so this is what this first step is, what luteinizing hormone is going to trigger in. So we saw this first step triggered by adrenocorticotropic hormone in the adrenal glands, but here in the testes it's triggered by luteinizing hormone. So remember, luteinizing hormone is what triggers the Leydig cells to make testosterone. So LH triggers that first step. And then the enzymes are present to make everything else. Okay, so then you have testosterone. Okay, and then from testosterone, remember we had to make DHT, dihydrotestosterone, and DHT was required for a few other things. So it's required for, we already learned that it's required for the external genitalia in a male, the penis and the testes, or the scrotum outside the male. So we learned that it's important for that, but it's also important for prostate gland development. It causes baldness. So we also see that. So there's an enzyme, okay, and you have to know this enzyme. It's called 5-alpha reductase, okay, and that's what converts testosterone to dihydrotestosterone, DHT. Okay. So it reduces something at the 5-alpha area. So there's another, so remember this one. Okay, so that's in male. So another one that we're going to come back and talk about later, but I did put it in here, is something called aromatase. So remember, testosterone is made here, so it can be made. Well, it can be made in the adrenal gland, but if the enzymes are present, but they're usually not. But then testosterone can be made, and then aromatase is used to make estradiol. Okay. So aromatase, androstenedione, which is made in the adrenal gland, if aromatase is present, it all depends on if this enzyme aromatase is present, whether or not these androgens are going to be converted into these estrogens. Okay. Estrone is an estrogen, estradiol is an estrogen, but this is happening in the ovaries. Okay. So we'll come back and we'll talk about that again. But these are two important enzymes that you need to know, aromatase and 5-alpha reductase. Okay, so going back to the DHT, okay, so target cells may be activated by testosterone or dihydrotestosterone. So in the body, you can see that there are a lot of things that are mediated by the testosterone. So differentiation of the epididymis, vas deferens, so we saw that during development, okay, increased muscle mass, puberty growth spurt, cessation of pubertal growth spurt, epiphyseal closure after growth, growth of the penis, deepening of the voice, spermatogenesis, and then there's the negative feedback and libido. So things that testosterone doesn't do, but is mediated by dihydrotestosterone, is differentiation of the penis, scrotum, and the prostate. So we talked about those external organs. male hair pattern, male pattern baldness, that's DHT, and sebaceous gland activity, and then growth of the prostate. So the reason, or one of the reasons we really want to talk about this, first of all, during development, if dihydrotestosterone isn't made or if the receptors aren't there, then there's no growth of the development of the penis scrotum, prostate. However, growth of the prostate is also mediated by DHT. So if we want to, if a man has benign prostatic hyperplasia, okay, or BHP, benign prostatic hyperplasia, or in other words, his prostate gland is enlarged, okay, that's being mediated by DHT. Okay? So DHT is contributing to the enlargement of the prostate. So the idea is that if we can block 5-alpha reductase, so use a 5-alpha reductase inhibitor, then we block the effect of DHT and we can stop the growth of the prostate. Okay? All right. And then, and so that's one of the big reasons why we need to remember this enzyme, 5-alpha reductase. Because the inhibitors are sometimes used to treat prostate gland, but they can the 5-alpha reductase inhibitors, finasteride is the most common, can also be used in a topical way to apply to the scalp. Men do this to prevent hair loss. Okay. All right, so pretty common. So hormonal control of spermatogenesis. Okay, so how are we going to control the production of sperm? So a couple of things. At puberty, secretion of gonadotropin-releasing hormone increases, and then it stimulates the anterior pituitary to increase secretion of luteinizing hormone, which remember we've already tied luteinizing hormone to Leydig cells, and testosterone. Okay. And then the follicle-stimulating hormone. I know this is repetitive, but I'm doing it on purpose. So Sertoli cells and sperm production. Sertoli, make of the sperm. All right, so FSH and, so LH and FSH, just by the way, FSH and LH are protein hormones. So we saw testosterone and the estrogens are cholesterol-based hormones. So let's see. So LH stimulates these Leydig cells to secrete the testosterone. Okay, testosterone is synthesized from cholesterol, so we're going back to spermatogenesis. Okay, testosterone is synthesized from cholesterol. It suppresses secretion of so the feedback mechanism is going to say, hey, we've got testosterone in the area, so the presence of testosterone, so we're going to talk about this first, feeds back to the pituitary and the hypothalamus to suppress more testosterone. So it's a typical negative feedback. Testosterone says, I'm here, look at me, I'm here, and the hypothalamus and the pituitary says, okay, you're there, so we won't, turn it up. We'll just keep it at this level. Okay, so continuous feedback. So testosterone, the other thing that it does here, without a lot of fanfare, but it needs a lot of fanfare, is it functions in a paracrine fashion to support spermatogenesis in Sertoli cells. So remember, the Sertoli cells are making the sperm. So they need testosterone to do it. So with low testosterone, the sperm will not be made properly. Or, yeah, okay. All right, and I put this in here, but I've already kind of talked about it. The enzyme 5-alpha reductase converts testosterone into dihydrotestosterone in these Leydig cells, and some adrenergic target tissues are not responsible to testosterone, but are responsible to DHT. So we've already kind of discussed that. Okay. So we just mentioned the Leydig cells and the LH. So let's talk about FSH and the Sertoli cells. So follicle-stimulating hormones act indirectly on spermatogenesis. Okay. So follicle-stimulating hormone and testosterone act on Sertoli cells. to stimulate that spermatogenesis. So Sertoli cells are doing it. The sperm production also happens with the presence of testosterone, but it is these Sertoli cells that keep the testosterone in an environment that supports their development. Okay, or keeps the sperm in that environment. Development of sperm. All right, so Sertoli cells are where spermatogenesis is initiated. Sertoli cells form a barrier. Okay, so this is an easy, obvious test question. Sertoli cells form a barrier to prevent the mixing of sperm and blood. This is something that you have to be aware of, that there is the blood-brain barrier, okay, and we talked about that in astrocytes and how they prevent, just blood from, the periphery of just kind of, all of the stuff in there from mixing. It has to go through the astrocytes that was in the brain. Well, in this case, it has to go through Sertoli cells, okay, to get into the testes, okay, which is where sperm production is taking place. So this protects sperm from immune destruction. So there's one thing about the immune system, and that is it recognizes patterns. Okay, certain patterns of things that aren't supposed to be in the body, and one of those things is really a flagellum. Okay, so the immune system associates a flagella with bacteria. Okay, so we don't want the immune system involved, is basically what this is about. So we have this blood testes barrier, prevents immune destruction of the sperm, prevents your immune system from attacking and destroying sperm. Okay, so what else do Sertoli cells do? All the stuff we've kind of already talked about. They provide nutrients to differentiating sperm because the sperm are isolated from the blood. You can't just increase blood flow. You have to have these cells there to push the nutrients there for the sperm. They secrete fluid into the seminiferous tubules to help transport the sperm. They synthesize and secrete androgen binding protein. So androgen binding protein is just something that, remember, testosterone is kind of so these are our Leydig cells out here. Testosterone is kind of moving into this area. Well, the Sertoli cells will produce binding proteins, so somewhere for that testosterone to land. So to keep the testosterone levels high and consistent in there. That's androgen binding protein, also made by Sertoli cells. All right, so Sertoli cells, so here's the question. Okay, we've made enough sperm. How does the pituitary gland know that we've made enough sperm? Okay, so that's kind of a good question if you just kind of throw it out there. It's like, okay, well, when you've made just tons and tons of sperm and you don't need to make any more sperm, how does the pituitary gland know that? Because the sperm's not going to be swimming around in the blood going back and saying, hey, I'm here. So what happens is that Sertoli cells release something called inhibin, and inhibin is what feeds back and will inhibit the follicle stimulating hormone release from the anterior pituitary. So that's our feedback mechanisms for sperm control. Very important to understand that because it's not the sperm that's doing it, it is the inhibin. The other thing, by doing this and just inhibiting follicle stimulating hormone, so even if the sperm numbers are building up and you want to reduce sperm production, you don't have to also inhibit luteinizing hormone and testosterone. So you can keep testosterone levels high, but reduce sperm production. I hope that makes sense. So that's what inhibin does, released by the Sertoli cells, allows testosterone production from the Leydig cells to continue. So we're not stopping everything, we're just stopping the activity of the Sertoli cells by blocking follicle stimulating hormone. All right, so then transport of sperm. Sperm leave the testes through ducts. Semen carries the sperm. So semen is produced in the seminal vesicle, okay, bulbourethral gland, and the prostate gland. So 3 places where you are making semen. So remember, semen itself doesn't have sperm. Sperm is made in the seminiferous tubules down in the testes. That's where the sperm is made, but then it has to have something to carry it, okay? And that's the role of semen. Okay? So seminal vesicles. So if we'll go over here and find them, seminal vesicle here, kind of sits there under the bladder. And then you can see that it can release semen into the urethra. Okay? The bulbourethral gland, it sits down here and will also put semen into the urethra. And then the prostate gland sits right here. This kind of surrounds the urethra and can also push semen into the urethra. All right, so note, I have a note here actually about the prostate gland. So note that the prostate surrounds the urethra and the ejaculatory duct. Okay, so the ejaculatory duct here. Okay. So it kind of surrounds that area. And so benign prostatic hyperplasia, remember where the prostate gland enlarges, will kind of push against the urethra and prevent urine from flowing through. Okay, so urine has to flow through that prostate gland, okay, to move out. So if that prostate gland enlarges, then it's going to squeeze that and prevent it from emptying. So BPH patients oftentimes have difficulty emptying their bladder. All right, so sperm and semen are carried by the vas deferens. So this is from the sperm here, so from the vas deferens here. And then the left and right, so this only shows one, but the left and right vas deferens will converge at the ejaculatory duct, okay, here. So there are two, there's one coming from the other side, but they come together, and then it goes through the urethra and through the penis, okay, during ejaculation. So production of semen, so accessory glands contribute 99% of semen. Okay, so those are the glands we just talked about. So the seminal vesicles produce fructose, vitamin C, prostaglandins, clotting agents. So these are all things that are going to improve the survivability of the sperm. Okay, so prostaglandins I have here may make cervical mucus more penetrable and may increase peristaltic contractions in the reproductive tract to help move sperm. Okay, so this may have an effect on the female reproductive tract to kind of move things, to help move things through. So that's the seminal vesicle, and then the prostate gland has buffers. So the female reproductive system is a little lower pH, more acidic. And so this has bases. So vitamin C, citric acid are also available as antioxidants. Proteolytic enzymes to break down proteins, and then calcium. Okay. Bulbourethral gland, mucus, and then again buffers. Okay, to kind of to improve that pH. Okay, so this is another picture. See the testes. So the seminiferous vesicles are kind of all the way through there, and then the sperm moves out. It's stored in the epididymis. Okay, so this will enlarge if sperm builds up in there, and then moves through the vas deferens. So one thing to notice here, I put this little note here. The vas deferens can be cut or tied, but you can cut that, vasectomy. Okay, so you just cut that there. And then you still have semen production. Okay, so you still have downstream of that, you have the seminal vesicle, you have the prostate, you have the bulbourethral gland. Okay, here. So you still have all of the stuff to make semen. It's just that that semen will not have sperm in it. Okay, so the sperm will not move through. All right, so emission and ejaculation. So emission, accumulation of sperm cells and secretions of the prostate gland and seminal vesicles in the urethra. So you start to produce, the sperm will move up during intercourse, and then will kind of collect in this area. Okay, but then the actual ejaculatory process is that's so controlled by sympathetic centers in the spinal cord. So this is a sympathetic oh gosh, what's the word I'm trying to think of? Reflex. Okay, so it's a sympathetic reflex. So that accumulation in the urethra sends sensory information to the spinal cord. Then that's what is going to cause peristaltic contraction of the reproductive ducts coming from the testes. And so here, and then so that's going to cause peristaltic contractions to kind of push things through. And then the seminal vesicles in the prostate, those will kind of push the semen into that area, and that's what the ejaculatory process is. So sympathetic and somatic motor input. So sympathetic, constriction of internal sphincter of the urinary bladder. We already knew this, actually, if you recall. So remember, during sympathetic activation, it's a sympathetic reflex that causes your bladder to, the detrusor muscle to relax and your bladder to fill. So we already know that sympathetic reflexes will cause closure of this so the bladder can fill. Okay, so we have to know that anyway. But it also happens during ejaculation. So constriction of the internal sphincter of the urinary bladder, so semen and urine don't mix, or so semen doesn't go up here into the bladder, or that urine doesn't come out. Okay. And then somatic motor, skeletal muscles and base of penis causing rhythmic contractions that force semen out of the urethra, and that's the actual act of ejaculation. Okay. And then the associated orgasm, that's a brain thing. Okay, so that's sends signals to the brain and release of endorphins. All right, so then last but not least, erection. So in erection, the physiology of that requires inhibition of sympathetic. So we just saw that sympathetic activation occurred as the ejaculatory reflex. Erection itself requires inhibition of the sympathetic activity, so parasympathetic is more dominant. Okay, it's not that important. I mean, it's important, obviously, but it's just different. Okay, so sympathetic. So erection is parasympathetic, and then it's a parasympathetic reflex, and ejaculation is sympathetic. All right, so medications that increase arterial vasodilation through maintenance of nitric oxide will help with achieving erection. So what's happening basically is that you have increased blood flow, okay, to the corpus spongiosum, cavernosum of the penis. Okay, so the penis will fill up with blood. Okay, so it's got this spongy part in there, and so these are supposed to be blood vessels. So as blood moves in, it will cause the penis to get larger and more stiff. Okay, so it's like filling up something else, I don't know, with something with fluid that's going to cause it to be more, firm. All right, so that's what happens. So we need to get that blood flow. Okay, so when we talk about medications, increase arterial vasodilation. So during an erection, these will vasodilate, increase blood flow into the penis, and that's going to cause it to fill with blood. Okay. Now, one thing that happens also is that it will compress veins. Okay, so veins that are draining the blood will be closed, and those will kind of be clamped shut because of the increasing in the size and the pressure will cause the veins to not drain as much blood. So it's kind of a positive feedback mechanism. The more filled with blood the penis gets, the less that blood's able to leave, which means it's going to stay erect. All right. So medications that increase that arterial vasodilation through the maintenance of nitric oxide. So nitric oxide is a vasodilator. Help with achieving erection. So something called phosphodiesterase 5 or PDE5 inhibitors will maintain high levels of nitric oxide, which is going to keep blood flow, help blood flow to the penis. Now, note I'm not saying that it's causing nitric oxide release, but it is maintaining a nitric oxide release. So when that nitric oxide is released as part of the erection process, this will say, okay, we're going to support that. Okay, so that's why you get the erection. It doesn't necessarily cause an erection, but it will make it much more responsive. Okay, So sildenafil, which is Viagra, and then, yeah, there are others. Okay, and these are also used for pulmonary hypertension because it also causes vasodilation. They were originally started out as research for cardiac medications, but it was found that they had this effect of increasing erections in men, and so they figured they could make more money with that, and they did. All right, so positive feedback mechanisms help maintain the erection. As the penis fills with blood, the veins are compressed, which prevents blood from draining, further maintaining the erection. So this is the flow chart: descending central nervous system pathways triggered by thoughts, emotions, and sensory inputs such as sight and smell. Input from mechanoreceptors, so touch. That's going to stimulate neurons to the penis, so increases activity of neurons that release that nitric oxide and decreases activity of the sympathetic neurons, which is going to allow blood flow, dilation of the arteries, the erection, and then the compression of veins is going to help maintain that erection. And that's it. Have a good day.